In this video, I am going to draw the anatomy of a bacterial cell. And the bacterial cell that I'm going to draw is going to be a bacillus, or a rod-shaped bacterium. So to do that, basically you want to draw sort of a hot dog shape. That's our bacillus shape there. And so this here is going to be our plasma membrane. All cells have a plasma membrane. All cellular life is made of cells. So why should bacteria be any different? They shouldn't, so they aren't. So another thing we're going to find in our bacterial cell, something we also find in all bacterial, all cellular life, is a squiggle here. This squiggle is going to represent the chromosome, or the DNA molecule. And the DNA is typically found in a particular region of the cell. It's typically all packaged up and bunched up into a region called the nucleoid. Nucleoid means it looks like a nucleus, but it doesn't have a membrane surrounding it. It is just in the cytosol, which is just the cell sauce that is found within the plasma membrane, full of all the proteins and the ribosomes and everything else, the aqueous solution that kind of keeps the cell running. So the nucleoid is this unbounded region of the DNA found inside the cytosol. Now, the nucleoid contains all the DNA necessary to do the basic running of the cell, we call housekeeping genes. Everything needed to reproduce this cell is found within that nucleoid. But sometimes we're going to find that there's going to be additional enhancements, genetic enhancements that can occur in bacterial cells. But we'll get to that in a moment. So another thing we need to have is, I'm just going to draw a few, but there's going to be like hundreds of these floating around in the cytosol. And these things with two components, a large and a small component, or a large subunit, a small subunit, these are going to be ribosomes. I guess I said ribosomes, so I'll point at two of them. So we've got ribosomes, then the purpose of the ribosomes is for messenger RNA translation, or when the DNA gets copied to make a messenger RNA, the ribosomes are what decode that messenger RNA in order to produce the proteins for whatever it is that this cell is going to do. Okay. As I mentioned, there may be additional loops of DNA that contain genes that are not housekeeping genes, but maybe something else that is going to be useful to the cell. Like if the cell is growing in where there's an antibiotic present, having some sort of antibiotic resistance is not something that every cell has or needs, but if there's an antibiotic around, it becomes very important to have. So the antibiotic resistance can be encoded in small loops of DNA called plasmids. So you might think of the nucleoid as being like the operating system for the bacterial cell, and plasmids are kind of like additional software, additional applications that can be they can be contained within the cell to enable it to do things that your regular everyday household cell wouldn't be able to do. So maybe they can metabolize a certain food source that normally the other cells wouldn't be able to metabolize. Antibiotic resistance is a common one. those kinds of things. So plasmids are these extra, much smaller DNA loops that encode genes. But it costs energy to transcribe and translate these plasmids. So if they're not useful to the plant or to the bacterial cell, it's gonna kick them out, or it's not gonna be using that plasmid. Okay, so that's pretty much what we find on the inside of the cell, but there's a lot of interesting things that can go on outside of the cell. So I'm going to go around our bacillus and draw kind of a second line here. Okay, so something that we frequently find in bacterial cells is a cell wall. And in bacteria, that cell wall is made out of a material called peptidoglycan. Peptidoglycan is partly protein, peptides, and partly carbohydrate, glycan. So it is a matrix of protein moieties and carbohydrate moieties that together form a hardened wall. And we'll see that there's a big difference in how different types of bacterial cells produce their cell walls. We'll come back to that. Okay, so another thing that we might find, I'm going to draw a third line around our bacterial cell. In cells that are pathogenic or cause disease, they may possess another layer of polysaccharide or protein called a capsule. And the capsule enables the cell to stick to host tissues. So say this was a bacillus that causes throat irritation. So it wouldn't be streptococcus, because streptococcus would be a coccus, but say it's some other bacterium that's pathogenic in humans. Well, in order for it to induce pathogenesis, it needs to be in contact with tissues in the host, which means it needs to stick to them. So it can use that capsule to effectively stick to host tissues. Because when you're as small as a bacterium, gravity doesn't really work the same way as an effective way to kind of keep you anchored to a substrate. So that's one way that bacteria can adhere to a surface in order to survive and be in constant contact with that particular other organism that they're interacting with. Okay. So I'm drawing a bunch of lines here. And a common mistake that people make is when they see these lines, they may think, aha, those are cilia. They are not cilia. They can't be cilia because cilia are something that's produced by, that are driven by microtubules. Microtubules are a part of the cytoskeleton, and the cytoskeleton is something that prokaryotes do not have. So because they have no cytoskeleton, they can't have microtubules. No microtubules means no cilia. So these lines here are not cilia, but are something different called fimbriae. And I'm just going to indicate here that fimbriae are not the same thing as cilia. So what these fimbriae are they're essentially rods of protein, kind of similar to, in purpose, to the capsule, in terms of enabling a cell to adhere to a host's tissue surface. But rather than being sticky and gooey like the capsule, the fimbriae are kind of like Velcro, in that they can like kind of poke into the host and remain attached in that way. So fimbriae, capsule both have the function of adherence to a substrate or a host tissue or something. Okay, at this end down here, I'm going to draw a flagellum. And if you remember from Biology 1020 that cilia and flagella function both by microtubules, you might be like, hey, Dr. Zanzo, what the heck? You just told us that bacterial cells can't have cilia, so how does it have a flagellum? And the answer is the bacterial flagellum is structurally very different in how it's formed versus how a eukaryotic flagellum is. I always think of a eukaryotic flagellum as kind of being like a cat's tail, because a cat's tail, it can swish around because it's got bones and muscles inside of it. Those bones and muscles are covered by skin. The skin is covered by hair, and that's how a cat's tail functions. Well, for a eukaryotic flagellum, the analogous structures to the bones and the muscles are microtubules and cross-linked motor proteins like dynein. And then the entire flagellum is covered by plasma membrane, which is analogous to the skin of the cat's tail. Well, it turns out that the flagella in bacteria is nothing like that. So I think of a bacterial flagellum as being more like a weed whacker. And what I mean by that is you've got this shaft to the flagellum, which is essentially, it's just a shaft of protein. So it's made up of protein subunits that aren't cross-linked in any way. Essentially, it's like a filament or a thread of protein. And at the base of it, there is a little motor, a little cellular motor that spins the flagellum around, kind of like a weed whacker. And this motor is very similar to what we saw in ATP synthase in the mitochondria. So, and it's thought that there's a good reason for that. That's by homology, not by analogy, that these motor proteins have a number of different functions, including making things like ATP synthase. Okay, so bacteria do have flagella, but they're very different. They're as different as, you might look under a microscope and see a flagellum flipping around and thinking, well, is it a bacterial flagellum or a eukaryotic flagellum? Well, eukaryotic cells are typically much larger anyway, but they might look similar, and they might have similar functions in terms of motility, driving this bacterium to somewhere else, but they're as different on the inside as a gasoline engine and a diesel engine. So, if you put gasoline in your buddy's diesel truck, he's not going to be very happy with you because you've done a lot of damage, and it's not going to run properly. So similarly, bacterial flagella, eukaryotic flagella do not run in the same way. Okay. So that's a bunch of the stuff on the outside of the cell. Now I'm going to draw something that's going to make this cell unique, in the sense that you would never see something like this on an actual living cell, which is going to have two different types of cell walls on it. I'm just doing this to demonstrate how bacterial cell walls can vary. And I'm going to use some color to illustrate this. Okay, so I'm just going to randomly select a couple of colors here. All right, what do I have here? Got this color, mulberry, and I've got this other color, true blue. Okay, so this outer thicker layer, I'm going to color that in the mulberry color. And this inner layer, I'm coloring it in the true blue. And I'm just going to indicate over here what those colors mean. Okay? So the blue means plasma membrane. Plasma membrane. And the mulberry means peptidoglycan cell wall material. Okay, so in gram-positive cells, they look like this. This is the structure of the cell wall. In most gram-negative cell walls, they look a little bit different. So I'm going to indicate that down here. So the typical arrangement in a gram-negative bacterium is they do have a cell wall, but that cell wall, like I've got here, is surrounded by a layer of plasma membrane on either side. And while I'm coloring this in, I'll keep talking about what is happening here. So the Gram stain, discovered by a microbiologist named Gram, he discovered that different bacteria will pick up stains, coloring agents, differently depending on the composition of the cell wall, which he didn't necessarily know that, but it was helpful in diagnosing what type of bacteria were coming back in different types of cultures that he was producing. So it's in some important pathogenic bacteria are gram-positive. So this is just like a first way to culture bacteria and then look at them under the microscope, stain them, and say, okay, well, which type of bacteria do we have here? Do we have gram-positives, gram-negatives? All right, so similar to here, there is a little bit of a space between the cell wall and the plasma membrane, but then we have this second layer of plasma membrane. All right, so in gram-positive bacteria, they are absorbing this crystal violet stain, and it makes the cells appear bright purple under the microscope. In gram-negative bacteria, the cell wall does not bind the stain because it's got this extra layer of plasma membrane protecting it, so they look more pinkish under the microscope. So this one down here is your most typical arrangement for gram-negative bacteria. So we've got peptidoglycan sandwiched between two layers of plasma membrane, or just one layer of plasma membrane and a layer of peptidoglycan here. You would never see a cell that has both of these things going on at the same time. This is just for illustration purposes. I want to make that very clear to y'all. Okay, so we've got these plasmids here. There's just one more little detail I want to draw in here, which is that these plasmids, these little loops of DNA, can be exchanged between bacterial cells. And I don't have enough room to make it in the same, I'm using a little bit of perspective here. So here's another cell. Another bacterial cell. And that plasmid of DNA can actually pass through this structure here called a pilus. Or if we have more than one pilus, we would call them pili. And that plasmid can pass through that pilus, a copy of it can pass through that pilus into another bacterial cell And now this cell has picked up that whatever information is on that plasmid. So if it's antibiotic resistance, hey, now this bacterial cell is also going to be able to be antibiotic resistant and survive. And this process of using pili to transfer genetic information from living cells to other living cells is called conjugation. And sometimes pili are called sex pili because we're increasing genetic variability, but it is not through actual sexual reproduction. That's something that's exclusively in eukaryotes. So this is kind of like describing it as sex in any way is kind of well, it's wrong. It's not correct. It's not accurate. But you'll hear it sometimes. Okay, so just one more note I'm going to add in here before I wrap up this drawing. So if our outermost layer is peptidoglycan, cell wall material, we have a gram-positive cell. Okay, if the outermost layer of the cell is not peptidoglycan, those cells are described as being gram-negative. Why am I repeating this? Well, because it turns out there's only one way to be gram-positive. The gram-positive bacteria form a clade because they all share that derived characteristic of having this arrangement of the plasma membrane and the peptidoglycan cell wall. Now, some bacteria may have, there's three different ways you can be gram-negative, okay? One is you have this arrangement where you've got two membrane layers sandwiching the peptidoglycan layer, which here I'm abbreviating as PG, the peptidoglycan layer. Okay, that is one way that a bacterial cell could be gram-negative. Now, if you're just staining cells, it could be that there's some other cell wall material which is not peptidoglycan. For example, in archaea, they have pseudo-peptidoglycan. So they're not going to bind the crystal violet in the same way. So that's a second and different way to appear gram-negative under the microscope. A third way, and this is true in some bacteria, including some that are actually rather closely related share recent common ancestry with the gram-positives, is just to have no cell walls. There are some bacteria that have no cell walls. There's some very important pathogenic bacteria that have no cell walls. So, for example, diseases caused by a group of bacteria called mycoplasmas. They have no cell wall. If you look at it under the microscope, they kind of look like tiny little blobs because they don't have the structure of that kind of peptidoglycan coating. So that's three different ways that you can be gram-negative, and only one way you can be gram-positive. So it turns out that gram-negative is polyphyletic, using some of our new terms. Whereas gram-positive, there's only one hypothesized ancestor of the gram-positive bacteria, and all of the descendants and only the descendants of that gram-positive bacterium have this this anatomy. So that is monophyletic, which means the gram-positive bacteria form a clade. So that's just me kind of trying to get us a little more comfortable with using these terms, giving you an example of how these terms can be helpful in trying to infer what the evolutionary history of different groups of living organisms are. So that's all I got for you for this particular illustration of our bacterial cell.